So thanks for staying up all day, and I promise I'm not too harsh. Let me try to tell you a story, actually. It's late in the day, so we're not going to start proving something seriously. Let me try this. So imagine you want to understand an outbreak, flu, whatever it is, Ebola. You want to know what happens at which time step. You want to know reliably. You want to know what is it you're chasing, right? So I heard this below the skin and above the skin, and my interpretation would be that some kind of microevolution happens in the body of the host as the disease is being transmitted over some kind of social network, right? So if we can agree of this being a reasonable scenario, I would be facing to explain to you as a mathematician how such an evolution of a virus is going to happen or could be modeled. That's one thing. The other thing, because I'm already outclassed by Madhav and DSSL in making those simulations, I'm not trying to go there. What I could do, though, is I could offer to help when the general comes with his question, right? How would you validate? And I'm trying, following my red line, where I want to talk about the spread of this disease over the social network. I'm trying to address this because all that mathematicians can do, we're not data-driven, not at all, but we're driven by structures. So what we try to identify are structures that help us understand deeper. And maybe I can get this across. So first, up, down, is that?
Keyboard forward arrow.
Here we go. All right. Okay. So let's go to the microevolution in the host. So obviously, let's say we have an RNA virus. We know these viruses mutate. So these mutations will oftentimes be neutral, hence Moto Kimura costs the term neutral evolution. So, but some of them will not. Now, we want to know, after a certain amount of time, where will the virus be functionally and structurally if we assume that there's a correlation between the two. So that's all obvious. What's not so obvious is how can I describe this? So what I have to do is I have to be able to create, say, a folding algorithm that makes from sequences structures. Now, there are two questions that come immediately to mind. And number one, this must be an accessible folding algorithm, so you must be able to use it with a nice interface and everything. Secondly, it has to be reliable. And thirdly, it has to produce something that we understand. So back in the 80s, Mike Waterman, who's fortunately here with us, created the same basic mathematics of a structural concept that we all use, namely that of secondary structures, right? And so 1978, right? Even further back. So these structures, we all use them in folding algorithms, the Zucker folding, RNA fold, Vienna RNA. There are many different versions of it, but it's a dynamic programming, very efficient algorithm folding these structures. So he has given us a concept of what we mean by structure. So I'm not quite content with this, and neither is he. So a few years back, we got together and tried to push the limits of what we mean by structure. So now you will say, well, I mean, now this guy is going to get really weird, right? But there's mathematically a precise way to talk about what we mean by higher structures, right? And this is what I'm trying to convey to you is important when I want to describe the evolution of this virus. Because this virus may not be confined in the secondary structures, because as you may know, in secondary structures, you don't have any cross-serial interactions. You guys with me still? So you need to push the concept beyond what Mike invented, right? And he was on board on that, and I'm very happy to have a paper with him on this, where we pushed those limits, okay? And this requires us to talk precisely about how we can push those limits. And this requires us to go beyond what we would typically do when we talk about networks and when we think of networks as graphs. So to go beyond networks as graphs means, in these pictures, and I just stick with pictures, it means that you replace an edge by a ribbon. So you pass to a belt instead of just a link in a graph. And by doing so, you can enrich the structure, eventually forming what mathematicians call a surface, which you then would say, why would he create a surface, right? For the love of God. The surface is going to give me invariance as a mathematician, and the invariance give me a grading about the structures. This is not so abstract because you don't have to be a topologist to get the invariance. You can read some of the diagrams in no time. So in literally linear time complexity, I can immediately compute all the invariants, and they give me a filtration of this structure space. So what this means is now that I have higher dimensional structures in the sense of more and more complex structures. I can fix a complexity level, and then I can look for the essence, as I was saying. It's not data that drives this, it's a structure. So it's how is the world of these structures organized? So the Waterman structures are essentially, as we discovered, right? They're essentially the base layer. They are the genus 0 guys. And then comes higher and higher ones. And we all know what genus 1 is, as we all know what a donut is, right? So genus 1 is a donut. It's essentially a structure that I can put on a donut drawn without intersection of the chemical bonds. And then it turns out that this space has an inner structure which allows me to reduce it to a very few configurations. So forget about the lemmas, that's all theory. But here are the only configurations that you can build on a donut. You see the 4 top configurations, and those configurations represent essentially the 4 different classes of structures that you have when you step a +1 over the normal secondary structures that we're looking at. So now with this picture, we can actually go into a database and we check whether we find this. Right? So we go into the RNA databases and we cross-check these structures, and we realize that, interestingly, a very high percentage of structures is actually just a concatenation and nesting of these motifs. Everybody with me? By that I mean that I take one of those structures, I expand one of the little intervals. I'm colorblind, so I probably will never see the light here. Let's see. Aha, not that colorblind. You're with me? I can go here, expand this interval, make it bigger, and squeeze one of the other guys, or the same, if I choose to, this, or this, into this interval. So I've nested the structure in itself. And that's clear that it's not going to do anything complicated to the structure. It's just a kind of repetitive way to look at this thing. So complexity classes, there are only very few. And once you fix one, you only have a finite set of configurations. So you get a structure of this space of virus realizations depending on mathematical invariance. And this gives us a way to establish the mapping. Let's go back. I said I'm interested in the evolution of, an imagined disease over time. And my task is to tell you where is the virus going in the host as time progresses. So I need to tell you which different structural configurations will the virus entertain, discover in the course of time. So you want to know, as a decision maker, will it become airborne, right? What kind of structural features will be preserved? And this line of mathematics can answer these questions, right? Now, then you will say, to establish the mapping, we need We need computer folding algorithms, and despite the fact that Madhav could probably do a much better job, and he probably will, we at least have proof of concept folding algorithms for these structures because we want to do something. So at the end of the day, when my guys are here, then we are going to have algorithms available for everybody who's interested in RNA and RNA folding, who will take sequences, I think up to 500 or 700 nucleotides, and can generate all structural configurations on a certain complexity level. So this is quite hands-on, and the basic building blocks of these configurations are the four archetypes that you hear depicted again. And we can compute the energies of those guys and can build beautiful recursive algorithms along the lines that Mike already established for the secondary structure folding. So I'm not going to tell you that there is slightly more math involved because it's late in the day to get these things recursive. But the type of mathematics that you can expect from us is very plastic. So we cut and we deform configurations in order to teach a computer to make those manipulations. In essence, instead of folding a pseudoknotted structure, we fold a normal structure and permute the base sequence later. So this is a fair way of looking at this. It's a clever a posteriori manipulation on sequence levels that can create the knotting we want. So now you're going to ask me for performance, and normally I would be very proud to tell you that this algorithm performs better than all the others, but we all have been in so many conferences where people make those claims. So let me try to make this, invert the statement. I'm not even surprised that the algorithm is better. And the reason why the algorithm is better than anything that is out there is because it's the only algorithm that actually takes into account the building principles. All the other algorithms do not. So in all other algorithms, you will always find a generic penalty for pseudoknots. But that is nonsense, because different ways of pseudoknotting have specific energies. And people are incapable of incorporating this because they don't have the underlying model. They don't have the decompositions. They don't have the unambiguous grammars that stand behind this. So the G-fold thing here, right, is actually fairly sensitive, and it has a very nice positive predictive value. And Phoenix Wang, who can't be with us, he's still struggling to get his visa, but in May he's going to be here. He's going to push this algorithm, so this is really nice and usable. And I encourage you to talk to us about incorporating this when you're interested in RNA folding. So we can push this a little bit further, namely when you are interested, suppose, right? So now I'm giving you a portfolio of things you might be interested in. If you're interested in interacting RNAs, we can use similar techniques to evaluate the likeliness of certain structures to form. Specifically, if you think that certain regions are going to bind, here you see these, I think it's red, but right? Be patient with me. So if you, if you have a particular focus on certain regions and whether they are being established in the complex, we can actually give you, using partition function methods, we can give you the probabilities of these regions. So you know where they bind and what type of binding will be there and how many nucleotides there are. And we can give you probabilities for these stacks to form. And here is a diagrammatic presentation which depicts the fact that we can have all sorts of interesting crossings even between the strands, right? Which is actually, to my knowledge, no algorithm can do this actually. This is something we can actually nicely do. And here is an example of a Homo sapiens snow RNA where you actually have these motifs. You have these specific crossings happening in nature, and they can be captured, and this is a nice topological model. All these algorithms run in polynomial time. They're fairly efficient, and we hope we can bring them and put them to use. All right, so this For us, the RNA is a paradigm. It's a paradigm as it is responsible. It takes the phenotypic form of the virus that I want to catch, and it is inside my body. And if I understand its evolution, I have to actually understand networks. Let me explain. So this particular RNA structure is going to be realized by many sequences. We all know this. All these sequences make the same structure. So the function is unique for all of them. What drives evolution is, say, a diffusive process of the genotypes that morph on these neutral networks. What drives the evolution of this process is actually how two of those networks intertwine in a very unphysical space, namely sequence space. So all the things that you've seen here are an integral part in this consideration, these evolutionary runs where you try to understand what is the virus going to do, when is it going to become airborne, how is it going to change. On a completely different level, I guess above the skin, we want to know what is going to happen as the disease spreads over a network. Now, again, networks. First, we had the microscopic network. Now we have the, if you will, macroscopic network. And that means, how can we decide whether me being node T at some location, after some time that people have become aware that an outbreak has happened, what's the probability for me to stay healthy? Fair question? So you may say that's not difficult, and you will want immediately to compute all the paths between, somebody whom you know is for certain sick and yourself. The problem is, well, the disease is not transmitting deterministically. There's a probability involved, right? And even though there may be many or a few paths, it heavily depends on how the paths are located on the network, whether I get sick or not. Well, let me try to explain. If all those paths have just one link, and if I were to remove it, and they all become disconnected, we all would agree I really want to remove that link, right? So it's not so easy. We are actually looking for independent paths. And my point here is a Gedankenexperiment that I want to invite you to join with me, right? This is what math is about, right? This Gedankenexperiment says, so we need to understand the independent paths in arbitrary networks, and that's usually very difficult. I think Stephen would agree with this on this one. It's very hard to do this. And here is a story I want to tell you about what math can do to help you to make progress on this one. So here, instead of looking at these paths, which are very difficult to understand and to control, what you could do is you could actually play a game. You could say, I'm not interested actually directly in the probability of me being infected. I want to hypothesize about the following. I want to hypothesize about the actual path of the disease in the sense of whom it infects first. I want to trace the causal path of the system until it reaches me. Now, you have to think about this for a second. When I do this, there is only one path from the infected person to me. You guys are with me? One path. Because I'm only measuring the guy who was infected first. There's no way that there's ever going to be a closed path composed by people who were infected first. It's impossible. So if I have that, then I actually have a tree, right? And instead of trying to count the number of paths from S to T that may be dependent and very difficult, related, correlated, right? I'm actually counting interpretations of the data that I'm being given. And the interpretations are subtrees of the data. And now these things are entirely independent because there's only one way this happened. I mean, right? There's only one way this happened. So only one of them is actually correct, and two of them has absolutely nothing to do with each other. You guys are with me? Right? So this means counting instead of the dependent paths, counting these realizations is completely independent, and all I can do is a combinatorial game, and we can certainly play combinatorial games. Let me assure you this. And this is a way where math is actually looking at a very well-known problem that can be computed, studying the corresponding processes on a graph. But you don't want to actually do this when you try to theoretically understand. Using this trick with the causal traces, the causal footprints that the disease leaves on a given network, you can actually compute all sorts of things, including very cool validation arguments. You can compute all distributions, and you can get a lot of information about the size even of outbreaks and the potential impact on you, right? So in math, it is important to identify structures that allow you to compute. It's not going to be a competition to Madhav. He can compute this better and faster. But what math can do is it can give you a structural understanding of the particular processes that at present can be so powerfully computed. And I think we have a paradigm shift because we can compute much faster than we can validate our computations, right? So here we're talking really about validation by a model. And a model is about ideas and not about data. And here in this case, it is about decorrelating paths that are hopelessly intertwined and that you could never control if you look at the problem from this perspective. All right? So I think we're going to have Andre give a quick. So we have 2 minutes until we hop into the panel.
Yes, but we can quickly.
Give him, yeah. Right. So rather than me talking all the time, I have
all right.
young Andre shoot.
So that was the story. Now comes the hard part. So this is a dynamic on a network. It's a simple dynamic. You have rules. The vertices change states. The topology of the network is fixed. For simplicity's sake, we work on Kn, which is the simplest, most connected graph. We work with a simple, unique probability of transmission. The states are susceptible, infected, and recovered. Normally, people would do that. They will give you the probabilities of transition for each individual vertex as a function dependent on the neighbors. One approach that we use, and he mentioned, is the decorrelation of the dependent paths between a source, a fixed source in the infection, and the test vertex. And you can do that by considering different realizations of the infection. So you can see to the right that suppose that the red vertices are infected, that the green vertices are susceptible, and that the blue vertices are recovered. One way the system could evolve with probability Pij, which in this case is just the constant probability P is the one above, and with complement probability it could evolve to the one below, right? So the idea is to count all such configurations which are strictly independent from each other, since each configuration, which is a realization of the way the disease spread, is unique, because we choose to say that the disease spread this way or that way or any other way. So the idea would be to move from a discrete dynamics standpoint in the network dynamics realm to structural motifs and counting trees and structure and combinatorial structures, which we can do with a fair degree of speed. As an illustration, we have one particular process which is called a CAR process, which can be interpreted as a terminated Galton-Watson process. This one actually solves the question of how big given infection would be at the moment it terminates, and this, perhaps without knowing it, what this process does is actually select such a configuration and count the number of vertices in the realized configuration and average over all of them. So the key question when we actually made these slides was, what is the distribution of the number of the number of new infected individuals at each time step. So what does the infection wavefront look like in a network, in a KN network with uniform probability? And we think we actually have a solution for this by counting these independent trees. So that's no longer a key question, but it is a cool question nonetheless. Obvious improvements to this toy model would be to now consider arbitrary networks, not necessarily KN, and arbitrary probabilities of transmission for each edge, which will actually give the model a lot of oomph when it comes to simulating diseases. Now, the key feature of all of this is the fact that we use mathematical inference. We don't use computation, so we use analytics. We get formulas, which you can plug into a computer and have it compute the numbers that you're interested in directly without having to simulate the system a given number of times and then compound statistically significant data. So that's our contribution. Thank you.
So if we have any questions, please, now's a great time to.
So how do you, I'm talking now about the molecular-oriented graphs for a moment. How do you see these new sorts of techniques being used for either the questions that we currently ask, or what questions do they allow? Do you see these tools potentially opening up? You know, if they were available in Patrick, if a tool that was based on this would pop up in Patrick, you know, how do you envision that they might be used by a biologist?
Okay, so the ad hoc use is folding and inverse folding of RNA sequences. And by that I mean, if you're interested in a short RNA interacting either with DNA or with another RNA, these tools can give you configurations, contact configurations of the RNA binding, and give you structural understanding, but no spatial embedding. So none of these algorithms are really referring to a lattice or to some 3D embedding. Currently not, but they give you contact information. You know who's paired with whom. So this is one class, and this is the folding. This is a mapping from the sequences to the structures. There is a completely different inverted applications line, which is inverse folding. Say you have a certain shape of an RNA, and you want to create sequences that are likely to produce that shape. Or, in fact, you want to manipulate the shape of that RNA, and you want sequences that have a certain structural feature you want. Then these tools can actually compute the inverse fold, which is equally complicated but inverse. The inverse fold can create subsamples of potential candidates of these sequences efficiently. These are also polynomial time algorithms, and those are ad hoc applications for the molecular foldings.
How about predicting something that would bind to that RNA and knock out its function?
You mean a molecular binding, like having a short sequence of an RNA and like some kind of antisense type of binding? Yes, this is very much possible. And I mean, it's embarrassing enough. I mean, we have done this in a math department. We have barely been able actually to find the time to do the conceptual coding. So right now we push the coding to a form that this is more user-friendly and things like this, because it's a long path to make things interesting and applicable for you guys, right? So we're in the process of doing this right now. And I'm quite excited that we have a chance actually to also hear what it is we can do in order to make this more useful, right? Because they were basically designed to get a publication done. We needed to have a C code that would compile and run and produce the data we needed in order to show specificity and PPV values, but there's much more to those tools. So they have not been properly PR'd.
My concern, if you can knock out the function of a ribosomal RNA, you can affect pathogenicity of the organism, so.
Right. Right. So I think they're fairly powerful, and I think if you think, I don't even know whether that's thinking outside the box, but if you think of function, the patterns that you're actually looking for are entirely different. Let me try to make that point. If you go into this microcosmos where one RNA has a whole network, one structure has a whole network of sequences that fold that structure. Right? So there's an exponential number of sequences, all of which form this particular structure. And I can find very easily two sequences where not one nucleotide in any position is equal. They're completely antipodes. And they still make the same structure. But many of the searches are actually sequence based. So I think there's a real possibility to shift this to this functional genomics paradigm, where you actually look for patterns, but the patterns are given by the structure. And these folding algorithms are, of course, very important here. Is this clear? Or?
So what you said is that looking at the sequence alone, you can't find these properties.
Exactly.
Only looking at the sequence with respect to its functionality can you,
Correct.
and in this case it's shape functionality.
Correct.
Can you understand their equivalence? They're equivalent under shape.
Exactly.
They're not equivalent under
Correct.
sequence.
They're part of the same neutral network.
So that's what a neutral network is.
But
Okay.
as a biologist, I'm having a tough time to wrap around sequence context to that shape, right? So for example, if you take like, say, small RNA regulation of RNA, transcriptional regulation of RNA and RNA. So how do I go from that, because I know that a certain microRNA, because of the sequence context, it's going to go and bind to the 3' region or whatever it is for a mRNA and regulate whether a protein is going to be translated or not. So how do I go from that sequence context or that sequence specificity to that shape?
So I'm not trying to say that sequence content can be totally ignored. What I'm saying is that, of course, molecular interactions have a very clear sequence signature. Some of them are totally dominated by antisense binding, right? It has to be the exact sequence of nucleotides, and otherwise it will not work. But what I'm also saying, if you look at the normal transfer RNA, there are bazillions of sequences that are not coinciding in a single nucleotide that will fold this transfer RNA.
It's not clear. I mean, a hypothesis would be that any of those sequences, the ones that fold the same, would produce the same protein.
So they're structurally, on a
That's a
certain level, on a
hypothesis.
certain coarse-grained level, they're structurally equivalent. For the particular molecular mechanism, you may need this particular, short sequence of nucleotides to be present to make this work. I'm saying there are two sides to it, and we've been focusing a lot on the sequence level. And I'm trying to stress the point that there is a completely different level here, namely that you have this kind of more abstract patterns. They're not sequence, they're still patterns. You just have to generalize the concept of patterns a little bit.
So I have another question.
Yeah?
So, and this has to do with the more general network. So in the particular case you brought up was SIR, but so SIR is a kind of a reaction diffusion process over a network. But so the more generally interesting question is reaction diffusion. It could be spin glass style computations. It could be all kinds of different things over networks. And so do you think that this sort of approach, and obviously this is relevant for learning and adaptation and computation of things. So do you see these techniques as being relevant, quite generally, not just with the spread of the flu or something, but just dynamics over these networks?
Right. Well, this is multi-layered answer, right? On the one level, right? The RNAs are for me particular networks. That's really what they are, because I tend to draw them as diagrams. I put the sequence on the x-axis and then I draw the arcs in the upper half plane. You know, I'm German, right? I like to organize things. And so they're a network for me. But they're only particular classes of networks. So what I'm really interested in are interaction networks. And I want to understand interaction networks. And what this group is going to do is going to extract structural principles that allow me to describe processes over those networks in any way that makes sense. Right? And that I can guarantee you we will do. We are not going to confine ourselves to something that I particularly well do, or he particularly well does, or Mike. Because Mike is also going to learn new things with us if it is required. So this I can guarantee you, and it's not going to be anything that is not completely tailored to the particular problem. But what we really try to understand are interaction networks, not even SIR. Right? This is something that we've done over the last 1.5 months together with Andre, and we were kind of and discussing with Stephen and learning a lot from Madhav and Anil. So we thought that's a cool thing to get started and to make some progress, and we're kind of puzzled about the structure that we see right there already. So we're quite excited about the first interactions that have been going on, and come my molecular guys. I hope to see more of you to talk to us, and then we make it happen and get these folding algorithms maybe put to use by you in a meaningful way. I would be quite happy about that.
Great. Well, thank you very much for a very informative question and answer session and talk, and we very much appreciated.